Chapter 10, Mutations and Cancer. Okay, so by the end of this lecture and your reading, you should be able to explain how mutations can occur, define proto-oncogene and tumor suppressor genes, describe how cancer develops and how it is treated, as well as list and describe differences between normal and regulated cell division. Okay, so what is cancer? We've mentioned it a few times, and I'm sure you have some idea about cancer because it's fairly common, but cancer is basically just a disease of unregulated cell division. And we'll go over exactly what that means and how it happens here in this lecture. So a couple of the genes that your chapter focuses on are these BRCA1 and BRCA2 or BRCA1, BRCA2 genes. You might be familiar with these. These are the breast cancer genes that we are aware of. So as we talk about things like this, and as we're talking about DNA and even like 23andMe, and when you're looking at these STRs, these short tandem repeats in your genome, we, like I said, kind of know where some things are, right? We know certain genes that cause certain things that might cause cancer, that might cause certain diseases that, we know where to look for some things. We don't know where to look for quite a few different diseases and cancers. There's still ongoing research for all of this, but some of the more well-known ones are these BRCA1 and BRCA2. So if you have someone in your family that has breast cancer, it's had breast cancer before, odds are that your doctors would probably encourage you to get the test to see if you have these genes. Okay, so they're located on chromosome 17 and 13. And just because you have a mutation in your BRCA1 or BRCA2 gene does not mean that you will for sure develop breast cancer, but it does mean that it increases your risk. Okay, so you guys can look at the graphs that we have down here. So we have risk of cancer over here, and then you have breast cancer by age 70 and ovarian cancer by age 70. And this applies to some other cancers as well. But you can see the light pink here is normal BRCA genes. So pretty small percentage of people getting or having a risk of cancer versus a BRCA1 or a BRCA2 mutation, much greater chance, usually between 40 to 60+% for breast cancer, and ovarian cancer still a much greater percentage at 39 or 11. So how do these mutations arise? How do you get a mutation in your BRCA1 or BRCA2 gene? And there's lots of other genes that can potentially cause cancer or lead to certain cancers or diseases, but how do they come up? Your DNA is really, really good at replicating. It does it all the time. We have about, I think, 6 billion base pairs in our genetic makeup, and there's probably only like one mistake every, I think, 10 billion base pairs or something like that. It's pretty uncommon. But the thing is, we make trillions and trillions of base pairs over our lifetime as we're going through cell division. Again, depending on what part of the body, because some parts of the body go through it more often than others. But even though these mistakes are really uncommon, eventually they can kind of lead up to something that is not so good, right? And that's how mutations arise, and that's usually what leads to cancer. We also have things like environmental insults. So obviously you've probably been told these things before, but smoking, sunburn, different kinds of radiation, etc., all of those things can damage your DNA and can lead to further mutations. So when these mutations arise, they obviously aren't happening super often usually. So the reason why that's happening is because our cell division is really tightly regulated. And this is what our lab goes over as well. And you'll hear me in lab, I usually mention 3 different checkpoints. There are actually 4. The first one is a little bit more obvious maybe, but there are 4 checkpoints and it's important to know all of them. So we know cell division, we go through interphase, which is G1, S, G2. And then we go into the actual division phase, which is mitosis. We end with cytokinesis, where we actually split it up. And then we go back into interphase if we're going to keep going through cell division. So a cell cycle checkpoint, again, there's 4 of them here. It's just the mechanism that ensures that each stage of the cell cycle is completed accurately. So it's like your little checkpoint before you move on, right? So, and you'll see G sub O or G initial here. We don't usually refer to that, but basically that is our first checkpoint. and it's just checking to see that we have the growth factors present to signal cell division. So this one's a little bit obvious, right? If you don't have those growth factors, you're not going to be going through the cell division process. If you do have growth factors present that are telling you maybe to develop a certain cell, or maybe it's telling a stem cell to differentiate into a kidney cell, whatever it might be, if those growth factors are present, then we're going to go into interphase, right? We're going to go into G1 where we're kind of doubling everything. And at the end of G1, we're looking to see if there's DNA damage that needs repair. So we're looking at the DNA that we have and seeing if we need to repair it, because if we do, we don't want to move on to S where we're replicating the DNA. We want to fix it first before we replicate it, right? Because if we replicate mutated DNA or DNA that's damaged, then we're going to have some problems. So we go through that first checkpoint. Let's pretend everything's good, right? Then we go into S. We're replicating our DNA. And at the end of S, maybe even kind of into the beginning of G2 is usually probably where we put it. We're checking to see have our chromosomes replicated properly. So did we have a mutation occur here? Mutations could occur either, you could either have mutations already or you could have mutations within the cell replication, the DNA replication. So this is our second or arguably third checkpoint, is have chromosomes replicated properly. And our last checkpoint happens in mitosis, kind of right in the middle. And we're looking to make sure that our chromosomes here are properly aligned for chromatid separation, which is happening during anaphase. Okay, so those are our checkpoints, and they're really valuable because if there is an issue, we want to make sure that we stop, go back, fix it, and then continue on with the cell division cycle. If there's an issue and we miss it, then we're going to run into problems. We're going to run into cancer. Okay, and you might have heard this before, but you'll definitely hear it here now or in your book, but we have something called apoptosis, and that's programmed cell death. It's basically cell suicide. It's not always a bad thing. A lot of times it's helping us from keeping damaged DNA from being replicated further, right? So when we have cells that accumulate enough DNA damage to interfere with multiple checkpoints, that's when we get cancer. So oftentimes we'll try to lead it into apoptosis if we can, or sometimes it's not really an option that our body will go through. So again, kind of talking about these BRCA1 and 2 genes, you can see the little pie charts over here. So if we're looking at breast cancers versus all cancers in general, basically only a small proportion of cancers are really due to these inherited mutations in the cancer-associated genes. So you can see up here in the breast cancers, 3 to about 5% are from inherited mutations in BRCA1 and 2, and then 2 to 4 0.5% are inherited mutations in BRCA1 and BRCA2 as well. The 3 to 5% are in genes other than BRCA1 and BRCA2. And then 90 to 95%, which is about the same for all cancers, are just acquired mutations that we get in our lifetime. So that might be from environmental insults. It might just be, a perchance damage to our DNA or to our cell cycle checkpoint, whatever it might be. But the gist of it is, just because you have an inherited mutation does not necessarily mean that you're doomed to cancer. So your family might carry these genes, but it is interesting if you look at certain ethnic groups, they are far more likely to carry them depending on which ethnic groups you're looking at. And you'll see this with all sorts of different traits, right? It doesn't just have to be diseases or cancer. I think your book mostly talks about Ashkenazi Jews. And one big reason why you see this in ethnic groups is because a lot of times, especially in older times where you weren't able to travel as frequently or as easily, you were possibly inbreeding, but definitely breeding within a fairly small population. And so that leads to mutations building on mutations and mutations, because say you have a mom and a dad who both have a mutation, and then they get together, they produce offspring. Now it's almost guaranteed that their offspring are going to have mutations, and it's a very likely chance that those offspring will then pass it on to their offspring, especially if they were to produce offspring with someone else in that ethnic group or in that population. So you can see how in certain ethnic groups you see prevalence of certain traits or diseases based on mutations that they inherit, basically. And you can see that's one of my, I don't want to say favorite, an interesting example is if you Google the blue people of Kentucky. I think those are kind of interesting because it's basically that they have a blue tint to their skin, and it comes from a line of inbreeding within a small population. So there's a chance that you can inherit those mutations, and there's also a chance that you can acquire them, right? And there's lots of different components of the environment, our food, and even our cells that can cause mutations. So when we're talking about these, we're usually talking about something called mutagens. Mutagens are any chemical or physical agent that can cause damage to your DNA by changing the nucleotide sequence. Okay. We also have something called carcinogens, and you've probably heard of these. You've probably seen them, especially on all of the, pretty much on everything has carcinogens, unfortunately. So when you're, you'll see a lot of like California laws that are like, this is known to cause cancer in the state of California, because California is pretty good at labeling those. And those are usually signifying carcinogens. And carcinogen is just any substance that causes cancer. Important to note that all carcinogens are also mutagens. Okay, so it's known to cause cancer, which means that it is a chemical or physical agent that causes DNA damage. Okay, so make sure you know that all mutagens aren't necessarily carcinogens. Mutagens might not necessarily always lead to cancer, although it is fairly likely, but carcinogens will lead to cancer and they are also considered a mutagen. So keep that in mind. Some examples of mutagens are radiation, whether it's from the sun or even getting x-rays. That's why when they give you x-rays, the techs leave the room, and why they usually give you some sort of lead vest to put on over you. Also chemicals, pollution, pesticides, smoking, alcohol, char, if you're burning food. There's also infectious agents, which we might see with COVID. We don't know. There's not enough research on it. It's pretty new. But some viruses like Hep C, you'll see that. There's also cellular reactions. So some cellular processes can also produce mutagenic free radicals. So these happen naturally, and they can be further kind of caused, I guess, by environmental insults, but sometimes they can just happen naturally, and that's just kind of the luck of the draw, unfortunately. So how do mutations arise? So if you're looking at your Chapter 10 class activity, you can also, this is good practice, so you can look at Table 10.1 to see what these mutations actually are. But mutations basically alter the nucleotide sequence of DNA. So if it's changing the coding region of a gene, the corresponding protein may be dysfunctional. And the problem is if that protein that you're trying to make helps regulate cell cycle, then you might end up with cancer. So you can see like a normal BRCA1 protein synthesis, we have our DNA, then we're going to transcribe it and translate it, and then we're going to make a nice normal BRCA1 protein. Whereas if we have a mutation, like say an insertion frameshift mutation, which just means that we're inserting one of the, basically a new base pair. So you can see the new base pair that we're inserting in red. So now it's moving everything over a little bit and it's changing our transcription, right? Because now we're going from, what were we before? I think we were a G before, so now we're a T. So we're changing this now to UAC, which is changing our amino acid, which is then going to change our protein, which means that usually when we change our proteins, it just means that we have a non-functional protein. Sometimes that's okay, sometimes that's really bad. In the case of BRCA1, that's not usually a good thing, right? There's lots of different kinds of mutations. Insertion frameshift is one of them. There are also deletions. There are also silent mutations, which means a silent mutation means that you change one nucleotide, but your amino acid doesn't change. So silent mutations usually aren't bad. So again, one nucleotide changes and amino acid does not change, which means that your protein would not change either. So again, a mutation is just a change in the nucleotide sequence of DNA. Again, there's a lot of them. Look at your table in your book. I think it's table 10.1. It'll show you all the different ones. There's inversions, missense, deletion, insertions, nonsense, lots of different kinds of mutations that can happen. And it does get difficult to kind of see them here. Again, the example that we're looking at down here is a insertion frame shift, but there are other ones that can happen. And remember, we talked about alleles before. We're going to get into them even more in the next two chapters, but alleles are just different versions of the same gene, okay? And they are produced through mutations. So again, mutations aren't necessarily always a bad thing. Sometimes they're just kind of a neutral thing, like it's not going to really affect anything, especially if it's like a silent mutation like we were just discussing. But a lot of the time we think of mutations as a bad thing. And that's kind of when we get cancer, right? If that mutation affects normal functioning of the cell cycle, it results in cancer. So there's two different kinds of cells that we have that are really important in cancer development. The first one is proto-oncogenes. Okay, proto-oncogenes are go or divide signals. So these are kind of like the accelerators. Okay, we all have proto-oncogenes, whether you have cancer or not. Proto-oncogenes are not a bad thing, but we want a normal one, right? Normal proto-oncogenes are proteins are functional, and it just tells your cells when to go. It tells it, signals it to go into a cell cycle progression when those growth factors, so remember our first checkpoint, when those are present, it tells you to go into cell division, right? So we want that. So proto-oncogenes are just a gene that codes for a protein that helps normal cell division. The problem is, if they are mutated, what normally happens is they're permanently turned on. So they're just constantly stimulating continuous cell division. Okay. So normally we want them to promote cell division and differentiation, but if they become mutated, they become permanently activated. This is when we call them oncogenes, right? And that is when you have cancer. Okay, so you know if you have cancer, you go to see an oncologist, right? So oncogenes is cancer, proto-oncogene, everyone has, that's our accelerator, that's our go signal that tells us to normally divide and go through cell division like normal. If it's permanently turned on, that's where we call it an oncogene, and that is cancer. The other kind of cell that we have is called a tumor suppressor gene. So these are the brakes. These are the ones that say, hey, you need to stop and you need to repair. Right, so these it's code for proteins that monitor cell cycle progression. Again, if they're mutated or inactivated, what they do is they can possibly cause cancer. So normally these guys, a normal BRCA protein, let's say, if the DNA damage is, if it notices DNA damage, it's going to say stop, repair it. It can either pause the cell division, it can repair the DNA, or it can initiate cell death or apoptosis, right? Which is what we would want if we had something that was damaged, okay? If this becomes mutated, it can become inactivated. So the other one, proto-oncogene, we permanently turn on. So it just goes, and it keeps dividing and dividing and dividing, right? So you just have uncontrolled cell growth. This one, it stops it, and so your DNA damage is not going to be repaired. So you normally have uncontrolled cell division, and those cells are going to divide despite having unrepaired DNA damage. So you're just going to get additional mutations that accumulate with every single round of division, which leads up to quite a few sometimes. Okay, so this is how you can potentially also develop a tumor, right? And tumors develop in stages as mutations accumulate in a cell. And it takes more than a single mutation to cause cancer. You're not going to get cancer because you have a BRCA1 gene mutation. You need more than that. So say you have a BRCA1 mutation, that's okay. You can have that. It does decrease your BRCA function. DNA is not going to be efficiently repaired, and you'll probably get additional mutations as time goes on, but it doesn't necessarily always lead to cancer. So you can kind of think of if you have a BRCA1 or BRCA2 gene mutation or any gene that we know associated with cancer, if you have that mutation, you can kind of think of that as like a head start towards cancer, which isn't a good thing, right? But it doesn't necessarily doom you. So thinking of it as a head start as you know, most people, as you get older, you've had more time, right? You've had more time for cell division, so you've had more time to accumulate mutations, which is why you see cancer usually in older humans. It does happen in younger humans. Sometimes it's inherited, sometimes it's environmental, sometimes it's by chance through cell processes, right? But we tend to see it in older populations. And that's because over time, you just accumulate more and more. So if you have a mutation in something like your BRCA1, then that's going to kind of give you a head start on that. So instead of getting cancer at 55, maybe you'd get it at 40. It's not always that dramatic, just an example, okay? So say we have that mutation, and then our DNA replication errors aren't being corrected, so then they're eventually producing an oncogene. So now we have this mutated oncogene, it's overactive, it's allowing the cells to keep dividing and dividing and dividing, okay? So then we get to the tumor suppressor gene. So let's say that in addition to having a BRCA1 mutation, you also smoke. Not a great idea, but hey, we all have choices. So smoking mutates your tumor suppressor gene, and now the cells are failing to correct any mutations or enter apoptosis, our programmed cell death. So they're going to continue. You also have the oncogene, so you have nothing stopping you, and you have everything saying go, 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 go. You're just going to be getting additional mutations. That can permit tissue evasion, metastasis, new blood vessel growth. So this is when you get tumors, right? Sometimes tumors are cancerous, sometimes they're not. So a tumor is just a mass of cells resulting from uncontrolled cell division. And you might have a tumor that might be benign, which means that it's non-cancerous. Usually when you first get a tumor, you do want to go to the doctor just to check. They can do a biopsy and they can figure out if it's non-cancerous, which, you can take it out if it's something that'll bother you. Sometimes they'll opt to take it out just in case. But really the problem is if it's a malignant tumor, that's a cancerous tumor whose cells can spread over the body. So if you have a malignant tumor, basically what you're going to want to do is remove it as quickly as possible before it gets bigger or before you enter metastasis, which is the spread of cancer cells. So when you hear that the cancer has metastasized, that's bad, right? That means that your cancer cells are spreading. It might just spread to a different neighboring organ. It might spread all throughout your body, but that's why we want to catch cancer as early as possible. So for treating cancers, I'm sure you all have heard of some of these. One of the most popular ones, obviously, is chemotherapy. Chemotherapy is treatment by very toxic chemicals. It's a full body chemical treatment. So the problem with this is chemotherapy doesn't target just cancer cells. Chemotherapy basically kills off everything. And that's why when you're going through chemo, you lose a lot of weight, you feel really sick, you have no appetite, you lose your hair, because it's killing everything. Obviously, it is relatively effective for most humans in treating cancer or letting you go into a remission, but there are definite drawbacks to it as well, right? So it's kind of an all-over treatment. It doesn't specifically target cancer. We also have radiation therapy, and that uses ionizing radiation. It kills any exposed cells by severely damaging the DNA. Okay, and that's kind of the same thing as chemotherapy. Again, it's not specifically targeting any cells. You can target more specifically in the correct area. So say you have prostate cancer, you can target that area versus the whole body. But a lot of times you'll see chemo and radiation coupled together. And again, they are really effective sometimes. So the drawback to it is usually, the benefit to it is usually outweighing the drawback. But not always. And it's a hard thing to go through. It does make you feel really sick because it's not targeting specifically. So they also have targeted therapy, which is great. And it's depending on what kind of cancer you have, it's not always available, or depending on what stage you're at. But targeted therapy is specific for cancer cells, and it's not harmful for normal cells. So that's something specifically that's really, really good to be able to go for if you do have cancer. And basically, they're just exploiting weaknesses that are caused by these oncogenes or the mutated tumor suppressor genes. So as an example, we're looking at this PARP protein here that repairs single-stranded breaks in DNA. So what the targeted therapy does is it'll use inhibitors to block the ability of the PARP protein to repair the DNA. so that the tumor cell will die eventually because of all the DNA damage. Okay, so that's an example of targeted therapy. You can also use something like immunotherapy, which uses the immune system to recognize cancer cells. And these are kind of a lot of the up and coming treatments, a lot of the experimental treatments right now, because immunotherapy basically allows you to target certain areas. So some cancers can evade the immune system because they produce a molecule on their surface. So they have something basically that hides them. It's like playing hide and seek, right? So they have something that hides them, so that immune cells can't be signaled to leave that cancer cell. So basically with immunotherapy, what happens is you'll block that signal and you'll allow your own immune cells to be able to kill those cancer cells. Okay, so that's ultimately what we want. So immunotherapy is often very effective, and it's awesome because it doesn't target your whole body. Obviously, one of the things you always want to do is reduce your risk of cancer. This isn't meant to be a preaching lecture, but it is sort of a PSA, right? Don't smoke or chew tobacco. You ideally want to maintain a healthy weight. You want to stay healthy, get your vitamins in, eat correctly, get vaccinated, avoid harmful environments. Make sure you use things like sunscreen when you're out, especially here in Hawaii where our sun is pretty strong. Screen regularly for cancer. There's different ages where you start screening a little bit more. But if you know that a certain cancer runs in your family, you definitely want to get tested for hereditary cancers. Again, it's not a dooming result, but it means that you'll want to keep a closer eye on it. And maybe instead of getting a mammogram when you turn 35, you're going to want to get it when you start turning 25 or 30. Okay, so that is overall the lecture on cancers. Again, feel free to do that class activity on mutations if you would like. It is optional, but it does go through all of the mutations pretty well that are in your book, I think specifically on table 10.1.